in-depth analysis of the osmoregulation mechanism and the effects of nitrate and salt concentration on the physiology of goldfish, Carassius auratus. The process of sustaining life in bony fish living in an aquatic environment, particularly the goldfish, Carassius auratus, is one of the most typical examples of biological adaptation and complex homeostatic regulation. For an organism living entirely in water, any fluctuation in ion concentration, osmotic pressure, and the accumulation of metabolic waste in the environment directly threatens the stability of its bodily fluids. The goldfish, a freshwater species belonging to the teleost fish group, Teleostei, faces a constant challenge. Its living environment always has a much lower salt concentration than the salt concentration in its blood and tissues. This creates a continuous osmotic pressure that drives water into the body across epithelial surfaces and causes salts to diffuse out into the environment. To survive, goldfish have developed a sophisticated osmoregulatory system, combining specialized organs such as the gills, kidneys, and intestines, along with a network of ion transport proteins at the molecular level. Besides natural factors, in captive environments like aquariums, chemical factors such as nitrate concentration and changes in salinity act as crucial physiological modulators. Nitrate, although considered the least toxic among nitrogen compounds, can cause serious chronic consequences if it accumulates excessively. Conversely, salt is often used as a supportive therapy, but can become a stressor if not managed at the correct concentration. This report will thoroughly analyze the mechanisms of water absorption, internal and external homeostasis, and evaluate the impact of these environmental variables on goldfish health and metabolism. Mechanisms of water absorption and osmoregulation in freshwater fish and goldfish. Osmoregulation is the biological process that allows fish to maintain a precise balance of water and salts within their bodies, ensuring essential chemical processes within cells function normally. The goldfish is a typical freshwater fish, with body fluids, blood, having an osmotic pressure of about 300 m-osm per liter, while the surrounding freshwater environment is usually below 5 m-osm per liter. This difference makes the goldfish's body act as a hypertonic system, continuously attracting water from the surrounding hypotonic environment. Water influx across epithelia and surface barriers. Water enters the fish's body mainly through osmotic pathways at the gills and skin. Although the skin and scales provide a relatively waterproof protective layer, the gill epithelium must remain thin and have an enormous surface area for gas exchange. The gill lamellar surface area in a typical fish can reach 200 to 300 per 100 grams of body weight, creating a massive gateway for water osmosis. Goldfish do not drink water actively like marine fish because they already receive enough necessary water through passive osmosis. Any water entering the fish's mouth is primarily for respiration, passing water over the gills, not for hydrating cells. Controlling this water volume requires a tight homeostatic feedback system to prevent cell swelling and membrane rupture. Excretory function of the kidneys in a hypotonic environment. To handle the excess water continuously entering the body, goldfish possess a kidney system with long, well-developed nephrons. The kidneys of freshwater fish operate at high capacity to filter blood and produce a large volume of very dilute urine, sometimes equivalent to 30% of their body weight per day. The nephron structure of goldfish is specially designed to reabsorb most essential ions like sodium, potassium, chloride, and other minerals before excreting the urine. This process helps minimize electrolyte loss while efficiently eliminating excess water. The coordination between glomerular filtration rate and tubular reabsorption capacity is key to maintaining internal osmotic pressure. The contrasting osmoregulatory mechanisms between freshwater and marine fish demonstrate the profound specialization of internal organs depending on the salinity of their habitat. Ion exchange mechanism at the gills, role of specialized cells. The gills are not only respiratory organs, but also the most important center for ion regulation in goldfish. Because salts constantly diffuse out into the environment across the gills, goldfish must actively scavenge ions from the water to compensate. Mitochondria-rich cells, MRCs, and epithelial cell types. The gill epithelium consists of three main cell types: pavement cells, mitochondria-rich cells, or chloride cells, and accessory cells. Pavement cells cover more than 90% of the gill surface area and are primarily involved in gas exchange, while mitochondria-rich cells are where energy-consuming, active ion transport occurs. In freshwater environments, mitochondria-rich cells use energy from ATP to pump ions against their concentration gradient. This process is driven by the sodium-potassium ATPase enzyme located on the basolateral membrane of the cell, creating an electrochemical gradient that allows ions to move into the cell from the surrounding water across the apical membrane. Independent sodium and chloride absorption mechanisms. An important discovery regarding the goldfish, Carassius auratus, is its ability to absorb sodium and chloride ions independently through different ion exchange mechanisms. Sodium absorption. Sodium ions from the environment are pulled into the cell in exchange for excreting ammonium or hydrogen ions. This process helps the fish both obtain necessary salt and excrete nitrogenous waste while maintaining acid-base balance. Chloride absorption. Chloride ions are absorbed into the cell in exchange for excreting bicarbonate ions. The enzyme carbonic anhydrase within the chloride cell catalyzes the production of bicarbonate and hydrogen from carbon dioxide and water, providing a continuous supply of exchange ions. Research shows that sodium and chloride can be absorbed at very different rates, depending on the fish's physiological state and the concentration of these ions in the water. This demonstrates that goldfish can fine-tune their ion transport system to cope with specific deficiencies of each mineral. The SLC family transport proteins and molecular mechanisms. The transport of ions across cell membranes in the gills and kidneys of goldfish is carried out by proteins belonging to the solute carrier, SLC, superfamily. These are molecular machines ensuring homeostasis at a highly sophisticated level. SLC26 family, versatile anion exchangers. The SLC26 gene family encodes multifunctional anion transport proteins capable of moving residues such as chloride, bicarbonate, sulfate, oxalate, and formate. In goldfish, these proteins play important roles in chloride absorption and bicarbonate excretion. The structure of the SLC26 protein includes a C-terminal SDAS domain, which plays a decisive role in protein expression on the cell surface and regulating the activity of other ion channels, like CFTR. The expansion of the SLC26 gene family in teleosts through whole genome duplication events has allowed goldfish great flexibility in adapting to environments with fluctuating alkalinity and salinity. SLC4 and SLC9 families, acid-base and sodium balance. SLC4 family proteins are primarily involved in bicarbonate transport, including sodium bicarbonate cotransporters and anion exchangers. Meanwhile, the SLC9 family, notably SLC9A3, NHE3, is responsible for sodium-hydrogen exchange at the apical membrane of gill cells. Transcriptomics analysis in goldfish shows changes in the expression of SLC4 and SLC9 genes when fish face salinity stress or pH changes. For example, in an alkaline environment, the activity of these exchangers increases to compensate for ion concentration fluctuations and maintain stable blood pH. Rh glycoproteins also closely coordinate with this system to facilitate ammonia excretion simultaneously with sodium absorption, an efficient integrated mechanism between osmoregulation and waste excretion. This network of transport proteins creates a coordinated system that helps goldfish maintain extremely stable intracellular electrolyte concentrations. Effects of high nitrate, NO3, concentration in captive environments. In the aquarium ecosystem, nitrate is the final product of the nitrogen cycle through nitrification. Although nitrate is considered less toxic than ammonia and nitrite, its accumulation at high concentrations over long periods, chronic stress, causes profound negative effects on goldfish physiology. Blood toxicity, methemoglobinemia, and oxygen transport. The most important toxic mechanism of nitrate is its endogenous conversion to nitrite inside the fish's body. Nitrate enters the blood through the gills and can be reduced to nitrite, which then oxidizes the iron in hemoglobin to methemoglobin. Methemoglobin cannot bind or transport oxygen, leading to tissue hypoxia even when the water has adequate oxygen. This condition, called brown blood disease, causes the fish's blood to lose its characteristic bright red color. At high nitrate concentrations above 100 mg per liter, methemoglobin levels can triple, significantly reducing the goldfish's ability to tolerate physical activity. Fish will show symptoms such as gasping at the water surface, lethargy, and reduced recovery ability after exertion. Impact on growth and metabolic efficiency. High nitrate reduces the fish's energy utilization efficiency. A significant portion of energy normally used for growth and reproduction is redirected towards cellular repair processes and maintaining homeostasis under toxic stress. Nitrate narrows the gap between standard oxygen consumption and maximum oxygen consumption, meaning the fish has less spare energy for development. Studies on zebrafish and tilapia show that prolonged high nitrate concentrations lead to significant weight loss and histological changes in the liver and kidneys. In goldfish, nitrate levels above 50 ppm are recommended as the threshold where negative long-term growth effects may begin. Immune system suppression and risk of secondary infections. Chronic nitrate stress increases cortisol levels, an immunosuppressive hormone in fish. This disrupts the balance between Th1 and Th2 cytokines, making fish more susceptible to opportunistic pathogens like bacteria, viruses, and parasites. High nitrate levels often accompany poor water hygiene conditions where bacterial density is high, creating a disease spiral that quickly exhausts goldfish. Nitrate safety assessment. Concentrations below 20 ppm are ideal, with no recorded negative impacts. Levels from 20 to 50 ppm are considered safe, but may cause mild stress for juvenile or sensitive species. Levels from 50 to 100 ppm are a warning zone, with slightly reduced growth rates and the onset of immune stress. Levels from 100 to 440 ppm are chronically dangerous, causing histological changes and a clear increase in methemoglobin. Levels above 1,000 ppm are acutely toxic and can cause death in a short period. Effects of salt NaCl, concentration on metabolism and solvent balance. Salt NaCl is one of the most common additives in goldfish keeping, but its impact on fish physiology is a multidimensional issue, strictly dependent on concentration and exposure time. Reducing osmotic stress and energy support. The main benefit of salt lies in reducing the concentration gradient between the water and the fish's blood. When water salinity increases slightly, for example to 0.1 to 0.3%, equivalent to 1 to 3 grams per liter, the osmotic pressure difference decreases, thereby reducing the energy the fish must expend to pump water out via the kidneys and absorb ions via the gills. This saved energy, potentially accounting for 10 to 20% of total basal metabolic energy, helps the goldfish have more resources to cope with disease or recover from injury. Salt also helps stimulate the protective mucus layer on the fish's skin, preventing the entry of certain parasites and fungi. Tolerance threshold and limits of a stenohaline species. Goldfish are stenohaline fish, meaning they tolerate only a narrow range of salinity. Experimental studies have identified critical physiological thresholds. At concentrations of 0 to 6 parts per thousand, goldfish adapt well. There are no significant changes in growth rate, cortisol levels, or hematological indicators like hemoglobin and hematocrit. Energy reserves in the form of lipids and glycogen in the liver and muscles remain stable. At concentrations of 8 to 10 parts per thousand, clear signs of stress appear. Blood cortisol levels rise sharply, indicating an acute stress response. Fish begin to experience muscle dehydration, appetite decreases, and feed conversion efficiency declines. An interesting observation is that at all salinity levels above pure freshwater, goldfish tend to reduce daytime locomotor activity, perhaps as a strategy to conserve energy during osmotic adaptation. The role of chloride in inhibiting nitrite toxicity. One of the most important applications of salt in aquarium management is its ability to protect fish from nitrite toxicity. Chloride ions from salt compete directly with nitrite molecules at transport sites on the gill cell membrane. When the chloride concentration in the water is about 16 times higher than the nitrite concentration, the ion transport channels preferentially take up chloride and reject nitrite, preventing nitrite from entering the bloodstream and causing methemoglobinemia. This helps goldfish survive even during periods when the biological filtration system malfunctions and nitrite levels rise. Although chloride does not directly compete with nitrate in the same way, maintaining a low level of salt still helps reduce the overall osmotic burden on the fish, thereby giving it better resistance to the effects of nitrate. Internal homeostasis and modulating environmental factors, pH, GH, KH, and temperature. Goldfish health depends not only on nitrate or salt, but also on the integrated interaction of other chemical parameters in the water. Fluctuations in these external factors directly force the internal homeostatic system to adjust continuously. Role of carbonate hardness, KH, and pH carbonate hardness, KH, measures the concentration of bicarbonate and carbonate ions, acting as a buffer to maintain pH stability. For goldfish, the ideal KH level is above 100 ppm to prevent pH crashes, a phenomenon that often occurs in tanks with high nitrate accumulation. When pH drops to acidic levels, pH below 6, the permeability of the gill epithelium increases, worsening salt loss and increasing water influx. Furthermore, the combination of low pH and high nitrate creates an extremely dangerous synergistic effect, reducing the blood's oxygen-carrying capacity much more strongly than when these factors act individually. Maintaining a stable pH in the range of 7.2 to 7.6 is optimal for the efficient activity of ion transport enzymes. General hardness, GH, and divalent ions. General hardness, GH, reflects the concentration of calcium and magnesium. Calcium plays an essential role in strengthening the tight junctions between cells in the gills. A calcium-rich aquatic environment helps reduce the leakage of ions outwards and water inwards, thereby reducing the energy cost of osmoregulation. Goldfish require GH above 150 ppm to ensure adequate calcium and magnesium for bone structure and intracellular biochemical reactions. Magnesium is a cofactor for the sodium-potassium ATPase enzyme. Magnesium deficiency in the water can impair the ion-pumping ability of the gills, causing electrolyte balance disorders. Influence of temperature on metabolism and hormones. Water temperature is a key factor controlling the basal metabolic rate of goldfish. As temperature increases, the rate of biochemical reactions increases, fish require more oxygen and produce more nitrogenous waste, ammonia, and subsequently nitrate. Temperature also affects the secretion of growth-regulating hormones like GH and IGF-1. However, an excessive temperature increase combined with high nitrate levels will push goldfish into extreme stress, as warm water holds less oxygen while the fish's blood has a reduced oxygen-carrying capacity due to nitrate. Conclusion and implications for goldfish health management. This extensive physiological analysis of goldfish reveals a system that is both sophisticated and sensitive to environmental fluctuations. The balance of water and ions is a continuous energy-consuming effort, coordinated by internal organs and specialized transport proteins of the SLC family. Nitrate, although less acutely toxic, is a dangerous chronic stressor. It not only directly harms through methemoglobin formation, but also weakens the immune system and reduces growth performance. Managing nitrate below the 40 ppm threshold through regular water changes and maintaining aquatic plants is a prerequisite for a sustainable aquarium environment. Using NaCl salt can be a powerful supportive tool for osmoregulation, particularly in preventing nitrite toxicity and reducing energy costs. However, overusing salt above the 6 to 8 parts per thousand threshold will conversely cause stress and disrupt the solvent balance within cells. Finally, the stability of the overall chemical environment, pH, KH, GH, serves as the foundation for all physiological activities. An aquarium with good buffering capacity and rich mineral content not only helps goldfish reduce the osmoregulatory burden, but also enhances their ability to withstand toxins and pathogens, ensuring the fish grow healthily and achieve maximum lifespan in captivity. Future research on transcriptomics and metabolomics promises to further elucidate the sophisticated adaptive mechanisms of goldfish in response to climate change and emerging pollutants in the aquatic environment.